Monica, can you hear me?
Yes. Yes. Can you hear me?
Yes, I can.
Good. Good. Thank you so
I,
much.
I want to apologize for the delay. I hope it didn't mess
No
up
worries.
your
No worries.
schedule.
No worries. I'm in a company retreat in a farm close to New Hampshire, in Boston, and I asked permission to. I excuse myself, so we can do it. We can do it. I have at least 45 minutes now, so we
A
can
nice,
definitely.
a nice background that you have in your back,
Yeah,
by
it's
the
a
way.
farm. It's a farm with animals and all of that, and I love farms. I come from a place in Chile where we have a lot of agriculture and farms, and I love it.
I love
Should
that.
I share, or what do you want to do first?
Yeah, you can share and just for us to see if everything is okay.
Let's see. Can you
Yeah,
see that?
I can see it. Perfect. I'm just gonna make a really short introduction of you for our
Yeah.
audience. So it's a great pleasure to have with us here another dear friend, Professor Monica Carrasco, who now holds two appointments. She's associate professor at Universidad de Talca in Chile, and also she's a scientist at Sensorial Therapeutics in Cambridge, Massachusetts. So Monica graduated and earned her PhD from University of Concepcion, Chile. Then she moved to the USA, more specifically to Harvard, where she did her postdoc with Tom Maniatis. She continued working with Tom as a research assistant when he moved to Columbia University. And during this period, she studied neurodegenerative diseases and established several in vitro human models for these diseases. After, as an associate professor at the University of Talca in Chile, her research focused on understanding neurodegenerative diseases using these in vitro models, in particular the model for amyotrophic lateral sclerosis, also known as ALS. In her lab, Monica used human stem cells from ALS patients that can be derived into motor neurons and used to generate new models to understand these disease mechanisms. Now, Monica is in very new, exciting path. She took a new, exciting path in her career, and she's translating her research at Sensorial Therapeutics, where she's developing a platform for screening natural products using human iPS-derived neurons. So, Monica, thank you so much for taking some time to be with us today. And I'm really excited to hear you this morning.
Thank you, Christina. I am really happy to join you. I am so happy and excited to be here. We can do, I prepare a longer presentation, but we can make it shorter and we can go to the main parts of the dish. And so, as Christina said, I study ALS. ALS is a neurodegenerative disease that kills upper and lower motor neurons. And incredibly enough, when you lose the capacity or the motor neurons degenerate, you don't really have symptoms up until the point 60% of your motor neurons are already gone. So it's a very interesting profile. The body has the capacity to actually compensate for the loss of motor neurons. Now, the most famous, it takes a name in United States as a Lou Gehrig disease, because this was a baseball player who actually had the disease and had a high profile. So it's a disease that is progressive, it's fatal. The median onset of age is around 50 years old, but you will see, probably you will know someone affected by the disease way younger, way younger than that. Death occurs because the motor neurons that innervate the diaphragm retract. And because they retract, you can no longer breathe. And in an ideal situation, you can get connected to a ventilator and extend your lifespan. But in countries like ours, Chile, for example, the ventilator depends on having electricity 24/7, and that sometimes fail. And so usually the people who has to live on a ventilator are in high risk, at least in our regions, because of problems with power, because of pneumonitis or secondary infections. And so after they get connected to the ventilator, pretty soon they find their demise. There are so many options that people have tried to be able to see or understand this disease. We know that 90% occurs randomly, is sporadic, and 10% is inherited or familiar. And within the familiar inherited version of ALS, we have identified many different mutations, as you can see here. The first mutation identified, the one that was heavily characterized maybe around 30 years ago, was a mutation on SOD1. And as the years has passed and go by, we have found many other mutations, and I'm going to talk to you a little bit about one in particular that is a mutation on a TDP43 protein that acts as a repressor or of the transcription. So it's very interesting in terms of gene regulation. And so, but I would like to say something, and this is, if you remember anything about this talk, this is the slide you have to remember. How do we model a neurodegenerative disease in a dish? You just saw this fantastic research where you can take a human tissue or you can take animal tissue and you can see the complexity of it. In the case of these models that I'm going to show you, these are in vitro models with all of the disadvantage that an in vitro model has, because you're going to have a neuron. And for it to be reflective of a disease phenotype, you need to have several things in mind. So one of the things that we need to do is we need to generate a model that allow us to grow a large number of neurons, because we want to do transcriptomic, metabolomic, we want to do biochemistry, we want to try to understand the disease in vitro. And so we need large numbers of motor neurons, which is difficult, whether you do it from a mouse model or any other model. Human models are even more complicated, and the cells that got developed for this research only started in 2007. So it's kind of not so new now, but it's kind of new still on the landscape. The other thing that you need is, in order for you to observe the phenotypes, you need to have co-cultures with glia, because you want the cells to live longer, to have an aging characteristic. The human motor neurons that are coming from these iPS cells from a human biopsy may take up to anything between 4 weeks to 12 weeks to generate particular different types of phenotypes. So that's something to keep in mind when you're thinking about this model. And third, in order that we could manipulate the system, we need to have a co-culture system that allows them to be in close proximity, but we need to be able to separate them to do all of the biochemistry, the transcriptomic, and all of that. So my talk is very different from what you have seen on the meeting, but because it's very different, it may be more complementary to what you may want to see in future. So we started with a mouse model in which we had the transgenic model that carries the disease and a mouse model that carries the HB9-GFP so that we can have the motor neurons labeled in green. And so when we generate stem cells with this mouse model back in 2007, 2006, we were able to test this proof of principle concept. Is it a stem cell derived neuron going to recapitulate what a real neuron does? We didn't know. And when we did this, we discovered that if we take these cells and we differentiate it into motor neurons, and then we co-culture them with glia, which is the supportive cell of the brain, we could extend the lifespan of the motor neuron in the dish, and we could, in that way, we could study early neurons, then a little bit more mature, and then an age phenotype. So that was a big advantage of the system. Even though it's a in vitro system, it's a very simplistic model, we could still see the development of the neuron. And then after we have that, we start to analyzing the interplay between the glia and the neurons. And we discovered something very important, that it's a mark in this graph. All of the red lines on the graph is the survival of motor neurons that are being cultured with astrocytes or glia that carries the disease. And all of the lines that are blue on the graph shows motor neurons, different type of motor neurons that have been cultured, that are healthy motor neurons that have been cultured with glia that is normal. So we discovered that when you have a motor neuron that is sick, like in the case of ALS, that sick motor neuron is going to die faster if the glia surrounding it is also sick. So there is something that the glia is doing that is kind of like making it like the niche in which the cell is surrounded by. It's kind of toxic. So after that, many people try to figure it out. What is the toxic factor that the glia is secreting that somehow is killing the neurons? We try to do that. Many labs have tried to do that. It's complex because it's not only one toxic factor, it's probably many different types of secretions that are making this environment of this niche in the spinal cord that is toxic for those motor neurons. This is a panel of glia markers. No, they don't look very different. We have done also transcriptomic on them to try to figure out what is the difference with these toxic astrocytes. We've done electrophysiology on them. We know that the neurons that are co-cultured with astrocytes that carry the disease have more excitotoxicity. They have more amplitude on the events, as you can see here, and they also have more frequency. So the presynaptic and the postsynaptic is affected on these neurons. This is a summary of this data. I'm going to go really fast on that so that we can go to the last part of the talk. We've also tried to study mitochondrial abnormalities because probably you have heard, like in the space of neurodegeneration, one of the first things that fails is the mitochondria. So if you look at the disease progression of ALS in a mouse, you can go from day 1 to day 120 to see the full spectrum of the disease. And the mitochondria are the thing that gets to be affected already at 5 weeks into the life of the mouse. So it's always important to check for those phenotypes and see, am I replicating my disease in vitro? And here you can see that mitochondria plays a huge role on neurodegeneration, and it's like a sensor. It acts like a trigger. And this is something important because if you want to look early signs, if you want to correlate, like whenever the disease triggers something like a biomarker that helps you to see that, mitochondria is a good candidate. And we did a lot of EM in collaboration with Juan Carlos Tapia at the Lichtman, at the time that we did this at the Lichtman lab. And we figured out that the phenotype of this aberrant mitochondria is present on this stem cell derived motor neuron. So it's really reassuring, and it validates the hypothesis that from a stem cell, you are going to chemically differentiate them into a motor neuron. And in that motor neuron, you're going to replicate phenotypes of the disease. This is the way a wild type mitochondria looks like. And here you can see on the two left pictures, the way that a aberrant mitochondria looks like. There are backwash, there's a disruption of the external membrane of the mitochondria, liberation of the content into the cytoplasm that cause an array of problems for the motor neuron. If you try to quantify the mitochondria with a regular mitotracker, you can tell that those cells that are affected by the disease has problems in terms of the number of mitochondria. So the mitochondria are affected, the numbers are affected. How do you measure this on a way that is efficient for a high throughput assay? And that's the biggest question that we need to tackle today. So we know that in these stem cells, we can recapitulate the disease. We know that they have the apoptotic patterns that are being characterized in the mouse, even though these are in vitro cells. We know that they have increasing synaptic activity and that they have mitochondrial abnormalities. So that we can observe the whole array of phenotypes of ALS in vitro, which is something that we didn't really know to begin with. And this is something I want you to remember. Whenever you confront a model to study anything, just always look for that. Look for the phenotype, because without that, there's so many examples in literature in which you will try to study a disease and your model doesn't give you the phenotype, or it gives you only one from 5 or 10 phenotypes that are heavily characterized. So the more you're closer to your disease, the better. Then Yamanaka developed this system in which you can take a skin biopsy with a skin puncture, a very little amount of fibroblasts, and you can culture them on a dish so they reach confluency on the dish. And once they are confluent, you can add 4 factors that he characterized and developed and discovered. that has the ability to turn this normal somatic cell into a stem cell. The whole process, if you are very good at it, or if you have a facility that does it for you, it could take anything between 60 days and 6 months, from the time that you select your colony to the time that you actually characterize the colony. But it opens a huge field, because now you have a human cell from a patient that you could actually study and evaluate a particular disease. What people thought about it initially is like, if I can make new motor neurons out of your own cells, maybe I can transform these motor neurons like this, and I can re-inject them so that I can do what is called therapy replacement. Yeah, but that's difficult because, of course, it's the spinal cord, and we have many barriers to pass. It's difficult to inject on the spinal cord. It's difficult that the neurons graft in the spinal cord. It's very hard that those neurons, if they graft, can extend the axons outside of the spinal cord toward the muscle. And so there are many challenges. I'm not saying it's impossible, but we're working really hard to be able to see if that will be possible in future. The thing that you can do right now with these cells, like now, it's the idea of taking these neurons from the patient, comparing it to a healthy donor, and see what is going on with these neurons, why they are dying, what's happening in their metabolism, what's happening in their gene regulation. And more importantly, and that's the reason of my latest career move, that I'm moving into the world of drug discovery, you can actually evaluate on a human motor neuron whether a particular drug or natural product will be able to rescue a phenotype. And this is very important, because by now what we have normally, the normal pathway in drug discovery is to take a drug, do in vitro assays with cell lines, then go for animal experiments, behavioral assays, then move to the preclinic, and then have a drug. And all of that takes about average 10 to 15 years. And our population is getting older. There are many more diseases, and we need to try to speed that process. So the human-based models are a good alternative to try to speed up the process and screen molecules that will be maybe more useful on the human context. And it will be not the classic thing that you test something on the mouse and then you push to preclinic, and then the human response to the drug is completely different from the mouse response. And so we did the same thing. We cultured the cell. Instead of being mouse, these are human, and they carry a mutation that is present on ALS that is called TDP. It is in a protein that is called TDP43. And the interesting part of this protein is that it has a very good influence on the expression of genes on the neuron. So if we think that the disease is actually changing the pattern of gene expression, this mutation is a great candidate to evaluate that. And so we try it with different co-cultures to establish the system. TDP43 is going to regulate the gene expression. There are many characterized mutations in this protein, 300 and something, and it could be more now. And what we start doing is looking at the cells. So here you can see in this panel of images a control line at 7 days, where you can see that the cells and in the dish start to spread their neurites. And then at 21 days, you can see a much more mature phenotype, a more complex circuit already formed on the dish. When you do the same experiment and you look at what happened with sick glia on these human neurons, you can still see that the sick glia is going to accelerate the death of these motor neurons by about 30%. So that's really a good validation that we can actually replicate that part of the disease in these human cells. You can also see that they have aggregates, which is expected for a neurodegenerative disease. And every time, every step of the way, when I did this from the space of academia, I encountered the same bottleneck. How are you doing this high throughput? How can you really evaluate not one line from one patient, but let's say 15 lines, 100 lines? So that is always a problem in the field, because you see, if you yourself take a biopsy and make your own IPS line and generate your own motor neuron, that motor neuron is going to respond to your genetic background. So the motor neurons of the person that is sitting next to you, it's going to have a completely different phenotype. This is a big problem in the field. We can have human motor neurons, but they are so diverse. How are we making a baseline with this system? And the way to make a baseline is to make it high throughput. But how can we make it high throughput? And this is always the limitation. You can see phenotypes like these, which is the aggregates of the cytoplasm. You can see granulates of messenger RNA. You can see problems on the transport of the cells that I'm not going to show because we don't have time to show the transport between the axon and the soma, or the soma and the axon, anterograde and retrograde transport. We have TDP mutations that leads to a partial loss of the function of TDP43. So we can assume that there could be a role that is lacking here, a loss of function or a gain of toxic function. So this is something that we need to always think about when we are trying to characterize ALS disease models in vitro. Here is an image of the work we did in collaboration with the Taylor Lab to measure the speed in which this TDP43 vesicle goes from the soma to the axon and from the axon to the soma. And you can see that the disease impairs the capacity of those neurons of transporting things. This is more of that, but I know that we are tight on time, so we can have more conversations later offline. But the summary is that you have TDP43 aggregates, there is disruption of filament, there is an alteration on axonal transport, and you have different viability when you co-culture them with ALS. Now, when you look at other molecular or characteristic of these cells, you can also see that the autophagy on these cells is altered. And so here I want to show you a little bit of the data that we have on changes in the autophagy system. So P63 is completely upregulated on those neurons. And it's very striking. It's one of the strongest phenotypes. So we know that this P62 is a very strong indicator of altered stress response in TDP43 neurons. We know that it could be potentially used for screening. And we know that the most robust phenotype so far is TDP43, is this P62 upregulation on these mutants. We know that happens very early, 7 to 14 days. So it's as early as the mitochondrial aberration in vitro. And here I can show you a few examples of what happened on the human system, where you can see aggregates of TDP43 in the mutant cells compared to the control ones at 14 days. So it's early. We can have a early biomarker. And now the only thing that we need to do is to take this system and try to make it at high throughput. So in summary, TDP43 aggregates as early as 7 days. The aggregates are present at 14 days, but the difference is less evident, and they are super robust at 4 weeks in vitro. So we have kind of like the ontogenia of how these aggregates form. And this is another example so that you can see a higher view of how these cells look so different if you compare the wild type TDP43 with the mutant TDP43. So we know that we have this phenotype. We know that we have a disponibility of a high number of cells so that we can do transcriptomic data. And we actually try. We try to understand these phenotypes on this particular mutation of ALS, such as the aggregates, the disruption of the cytoskeleton, the autophagy phenotype, or the increase in expression of APP with the protein changes on the cells that are summarized here, and also with the idea of thinking about how these cells can be used to understand the gene expression. So we have a system that is slightly different from other systems in which we culture the glia and the neurons separately, and then we put them on co-culture, and they are in close proximity, but they are not touching each other. So with that, what we do is that we have a good system in which we can separate the cells, and we can actually do a transcriptomic experiment or metabolomics, or do an exome or secretomic analysis, so to understand not only the phenotype that the cell tells me, but something that I can put on a pipeline to understand it more on high throughput. And so we use several lines in Chile. These are the lines that we were using in our lab, ELA, Huntington. We also acquired some Parkinson lines to try to establish the lines, the different disease models for the cells, and we design a paradigm to do transcriptomic in a way that we can actually try to understand the interplay between the astrocytes and the neuron. I'm going to go really fast on this because I imagine transcriptomic, it has its advantages and disadvantages. One of the biggest advantages is that you can actually get a lot of data out of it and it be very specific. But one of the problems that happen when you do this type of analysis is that you really need to have a strong computational biologist background so that you can avoid the big data issue, which I'm going to talk a little bit at the end. These are the genes that are either enriched or downregulated on these TDP43 neurons. As expected, the enriched genes are associated to nervous system development, ion channel activity, and metabolic glutamate and GABA receptors. And the ones that are downregulated in the neurons are those that are allowing the neuron to connect to another. So it makes sense. It all makes sense. We know that there are a few aberrant genes that are like the usual suspect, things that we were expecting to see. And what we also wanted to know is if this protein TDP43 that alters gene regulation may have affected splicing. And we look for that too. And let me show you the summary of that. Here you can see how if you compare the splice happening on mutant cells with the splice happening with the wild type cells, you see that there is an increase on differential splicing, especially on the splicing that occurs through a cassette exon splicing. And you can see also the targets. And we have been exploring more and more what are each one of these top targets doing in our system. But how do we begin the screening? And I just wanted to make a pause here because I think this is something very, very important. If you have a model, how are you going to upscale your model? This is really like the million-dollar question. So I needed to learn how to do an high throughput model and screening. And luckily, Lee Rubin in his lab at Cambridge allowed me to learn, helping a little bit in this project. The project that was led by Richard Krolewski, and the idea of this project was to use high image content to be able to screen either different cell lines or different products on the cell lines. So what I'm going to show you here is a little bit of what I did here, just very high level vision. We had iPS cells, they were edited. You can differentiate it into motor neuron. We have 3 different lines that were at any given time analyzed. And then in this high image content analysis, you could actually multiplex and determine several different types of phenotypes. And the idea is that you can focus on many different lines at once. You can do a functional analysis on these cells, and you can do a high image content analysis on these cells. And at the same time, because of the way that these cells are and the way that you can increase the production of the cells, you can actually do screening for particular compounds. In this particular project, that is not my project, I was just helping to do a screen of pesticides. And this, if you want to remember something, whenever you need to learn something, it's always very good to just get involved in a project, even if it's not your main interest, just to be able to put your hands to it, to try to learn how to do it. And then we study this, we differentiate the 3 lines, and then we put it into a high image content to just see neurite growth. Just look at the cells, how they grow, how fast can we do this image content. This is a image content that has a format in which, obviously, maybe you have talked about it in previous talk, but it has a format in which you can put your dish and you can track the cells live, because it has CO2, it has temperature control, and you can do it in any different formats, 96 wells, 384 wells, and more. It's from molecular devices, and I am trying to learn several different types of these devices so that I can multiplex my tests right now. And so it's a very interesting system because you can have in one plate basically 384 experiments. 384 that could be either to test something or to test yourself. And I think that is powerful. That is really powerful because in the realm of stem cells, we have a lot of variability based, one, on the genetic backgrounds of the cells. So that for us to understand something that is relevant and valid, we need to screen many different cell types. And I wanted to point out the big challenges in this. And actually, Val talked about that at the end of the talk. How do you actually process this data? We have a big data issue. How do you interface the software of your high image content device with the data? How do you control that? Us that we live, now I'm transitioning to industry, but normally in our labs in Chile and probably in Brazil or Argentina, you get the device, you get a device that has these capacities and you go crazy for it. You want to try it, you want to test it, but there are so many issues about to solve. One of the things that I learned the hard way is that you don't want to get a device that you cannot interface, that you cannot generate a math lab code or a command line interface, because one of the bottlenecks of your analysis is going to be taking that data and process it in a very streamed way. You don't want to be sitting in front of the device doing 20,000 clicks to get each line of data. And the other thing that we need to think about when we are thinking about high throughput, at least in Chile, is the cost and the maintenance fees. Because one thing is to get the device, but the cost on the maintenance are high because they need to be very good in alignment. Now, I have the fortune to visit Universidad a couple of years ago, and I see the advantages that you have there. So you have a fantastic imaging center, fantastic. And you also produce your consumable. So in terms of cost and maintenance fees, you can push it down because you don't need to import everything. And that's a big advantage. I don't know if you have talked about it through the meeting, but this is an advantage that should be valued enormously. And so what I'm doing now is to try to use another type of device that is slightly more simple than high image content, but is very powerful. And the device is called InQSight. It has pros and advantages and disadvantages. I know there is one in Brazil because I asked the company, they have one in Brazil working. And what we are doing is we are putting the cells, we are loading them into the device, and we are actually looking at the bright field images to see how those neurons grow. And then the device can take the images, as many as you want, as frequent as you want, automatically. So the cells live, the device lives in the incubator, the cells live in the device, and you can image them to really follow on time how much they express their neurites. So here is an example of a raw image. Here is a segmentation. Just for visualization purpose, you can see the projections here, and you can see the data. And so you can actually segment these cells in a very fast way. This is done at the same time that the images are acquired. So you can follow over time a phenotype that will allow you to evaluate either neuroplasticity, neurodegeneration. You can do calcium analysis on it. I'm exploring now the functionalities for calcium analysis and for fluorescence on the machine. This is another example of the segmentation. This is another example so that you can see how the raw data looks like. So it's very good and very well fine-tune so that you can use all of the algorithms that are within the device. They give you a particular package so that you can analyze length or cell bodies and do densities. But one of the problems that I have is, of course, the data analysis. Again, we're going to go back to the big data issue. This device, and this is what I said before, I learned by knocking my head on the wall. This device does not have a good interface. So it's not an open software. You cannot do MATLAB code to stream the data. You cannot do command line interface to stream the data. And that has a huge time-consuming problem when you have to actually retrieve the data out of the device. And I think that's important to keep in mind, because like devices like Val was showing, when you use Imaris and when you use all of those systems, are more open. And you always need to look for something that, even if it's not so intuitive to use, it's very open for you to be able to use the data out of that. The work has been done in my lab by many different people. This is my guys. Can you hear me? Hello? I'm not sure.
Yes. Yes.
Yeah. Okay. This is my students. Daniela, my first graduate student. My first postdoctoral student. Alejandro. Francisca, she just entered a PhD program in Chile. Braulio, an excellent postdoc who accepted a position in New York, and my first undergraduate in medical technology who is currently working on the private sector. And I just want to give thanks to this is the work of a lot of different people. And of course, big thanks to Professor Cristina, because I think just starting this conversation, I know that you are expert on imaging. I know that, for example, what Val showed just now, it's incredible. The power of multiplex, different antibody recognition on a tissue, it's amazing. And so I really enjoy looking at this, learning from you. And I just wanted to let you know that whenever you confronted this, in whatever model you do it with the knowledge of the challenges that are going to be ahead. Okay, that's everything. I'm sorry, I just fly through it because we have very little time, but I'm happy to chat with you later on or maybe offline if you have more questions about it. Right now I joined a company. I'm helping to establish a platform to do drug discovery based on natural products. So it's a whole different ballgame, and it's very exciting. I think I was academic hardcore person, but I can tell you that the industry has worked.
Your microphone went out. Yeah.
Okay, there. Can you hear me now?
Yeah, perfectly.
Okay.
Thank you so much, Monica. Can you stop sharing your screen?
Yes.
For this very exciting talk. I just can't wait to chat with you, because there is so much that we can do together to collaborate. So it was a great talk, and many perspectives are open here, I would say.
Thank you. It's very different from what you have seen, though. I noticed that your expertise are very complementary. What you show on this meeting is very complementary. I feel like I'm doing baby steps here.
No, not at all. I think that one good thing is in this workshop is to show the diversity of techniques that we have, and all the speakers were so generous in sharing with us not only the good sides of the techniques, but also the troubleshooting. So it's great to see such different techniques with the different applicabilities. I don't know if we have time for questions.
Greg, I think Greg has a raised hand.
But I think
I just have one very quick question. I'm curious about when you separated the glial and the neurons in culture. Is that through some membrane, or is it just two opposing cover slips?
Yeah, that's a very good question. So I put the glia on the plastic and I treat the glass with wax. So I generate a tripod. It's an artificial tripod. So before I play the neurons, I put 3 dots of paraffin on the glass and I add the neuron. It's so much more cheaper than a transwell. And the neurons thrive on glass. They like the glass. They because it's coated with polydilysine and laminin. So they hook to the glass. And then I take the glass where I play the neurons and I invert it over the glia. So I can take the glass has the advantages, you know, when you do RNA-seq and you have to do trisol extraction or fast freeze, and you have to process the whole sample. Here, because it's on a glass, I can take whatever sample I want at whatever time I want and process it separately without making a problem with the rest of the dish.
Yeah, I'm just curious if you've ever looked, because I'm sure you know, like either called cytonemes or scanning nanotubes. So the long connections. So I'm curious if those, because those during development, they can grow millimeters. And so I'm curious whether you've ever checked just to be sure that some of that transport is being done by cell contact or through cytonemes, or if it's diffusing a material. I mean, you kind of think diffusion, but I would still like to know whether it's
I have not looked. I have not looked, and it's a fantastic question. Actually, it's a fantastic question. No, I have not looked at that carefully.
Yeah, it could be actually an interesting point to think about.
Yes.
Okay. All right. Thanks very much.
Thank you, Greg.
Any other questions? So Monica, if I may ask you, I don't know if you can answer, but what are you testing right now?
So I can answer the general version of that.
Not giving too much detail.
Yeah, without much detail, but we are testing psychedelics
for
the disease like anxiety and depression.
Mm-hmm. Yeah, which I think got worse after the pandemics, right?
Yes, definitely. And that's something really interesting because people have not moved that much into the mental space, which is some unexplored areas. So yeah, but the good thing about it is that once the platform is ready to go, it's going to be good for anything, not only for psychedelics, but for anything that you want to test on human neurons.
And are you using a platform that's similar to the one that you're using at Rubin's lab?
Yes, very similar.
Very nice. Unfortunately,
and hopefully for next time I'll have more data to show you.
Yeah. Go ahead, Greg.
Let me, Monica, I just want to mention 2 things. I don't know if you're familiar with the NanoLive microscopes.
No, I'm not.
Yeah,
so
I'll send you.
I'm baby steps on it. So
yeah, I'll send you a link. I think it could be interesting in exactly the type of thing that you're doing. So that's.
So I want to make a note. How is it called?
NanoLive. It's all one word together. It's made in Switzerland, but they have some neuronal cell differentiation, neuronal cell migration movement, all sorts of things with NanoLive. And so it's all done with, it's done without staining any of the cells. It's a new system. It's with essentially.
Is it with the nano antibodies?
No antibodies. You don't use antibodies. It's essentially a 3D imaging with reflected light with it's a hologram. It's actually what it's called. It's a hologram. So I think that would be something interesting.
Impressive. I'm making a note right now. I'm like typing it right now. I'm super interested in to see NanoLive microscope. Yeah, I'm going to research, and if you could please send me the information, I will be wonderful.
You'll find that. And also the second thing I'll mention is that in the CZI project, so CZI, they have another whole group of projects of computational big data. And so there's a whole system within CZI. It's called NAPARI, and so they have all of these projects for big data analysis. And I don't know specifically for neurons, but I would suggest that you also look at Napari, Napari, Napari.
Yeah,
Okay.
I'm making the note right now. Thank you so
Okay.
much, because if this is not my expertise, and I saw the talks, they were amazing. The talks today and yesterday. I mean, just so amazing. The expertise that you do guys have is incredible, and the autonomy that you have in Brazil, Cristina, it's also super valuable. I don't think that maybe you take it for granted because you have it there, but in places like us in Chile, we don't have the autonomy that you have in Brazil. That's great. So I'm going to research on your suggestions. Very interesting. I need to talk to Cristina because I'm eager to try natural products from Brazil.
You're just too kind, Monica, and we are looking forward to have you here with Juan Carlos visiting our facility. I don't take for granted. I'm pretty aware what the microscopy center here represents. It's a fantastic facility. We're very proud of it.
Yes, it is a really fantastic facility. Thank you so much. I have to run because I'm on another meeting, but I excuse myself because I really wanted to be here. And please, if you have any question, just shoot me an email if you're interested on what does it mean to transition from academia to industry. If you have any other questions, I'm super open to talk to you offline. So just, you have my email, so just go for it.
I will share it. Thank you so much, Monica. Abraços.
Okay.
Bye.
Besos. Ciao.
So I think we can close this morning section. And